G'day guys, Jeff and Matt from ATP Science. We've got a personalised health summit that we're doing at the Gold Coast Convention Centre on November 2nd and 3rd. Matt, what is your personalised health summit all about?
Well, basically what we're going to do is we're going to help you find out where you're at, go through your symptom picture questionnaire, find out what your particular health priorities are.
Yep.
And then over a couple of days, we've got some great guest speakers coming from everywhere, all over Australia and the world. We've got Rich Kreider coming over, the expert on creatine.
Head of the ISSN?
Yeah, he's a head of the International Society of Sports Nutrition.
Yep.
And works with Texas University, mainly the NFL and the American military, those sort of people. So some great knowledge.
Grandfather of creatine, actually, one of the guys.
Pretty much.
Yeah, I mean, he's a smart guy. Yep, who else we got, Matt?
Simon Hill from Plant Proof.
We got excellent, we had him on the podcast.
Fofi Van Kampen.
Yeah.
And she's doing some gardening and all that sort of stuff. We've got James Newbury, Cara Saunders. But
from CrossFit?
From CrossFit. So what you're going to do is we're going to go through, find out where you're at, what your particular health priorities are, and then over a couple of days we're going to have all these keynote speaking, these electives, so you can go and learn about your particular strengths and weaknesses and how to take that to the next level in regards to performance.
The thing about the speakers that we've got as well too, it's a real cross-section. Oh, don't forget, we've also got Liz Lambert as well too.
Ken Ware. I mean, he's capable of getting people out of wheelchairs. It's probably worth a mention.
Yeah, I mean, like the chaos theory. I mean, he was on 60 Minutes and literally have a look at some of the speakers we've got.
He's changed my life in 6 months.
So it's really going to be something that's a bit special, and you're going to be able to follow on with your own questionnaire so that when you're listening to the information, you're going to be able to effectively help to diagnose your issues or your areas and things that you want to be able to work on. So, and at the end, we're going to get all of these guys together with a big panel for a massive Q&A section. You're going to be able to break out into electives and ask questions and learn specifically about different areas that these guys are experts in. So it's something pretty different, pretty exciting, and we're looking forward to it.
Yeah, I can't wait.
Yeah. All right. Well, jump online to atphealthsummit.com. That's atphealthsummit.com, where you can actually learn more. Of course, you can always get in contact with us through Facebook, and also jump onto the websites. There'll be links there as well too. So, Matt.
Sweet. See you there. November 2nd and 3rd. Hey, g'day. Welcome to the ATP Project. You're with your host, Matt, today, and I've got my good mate, Diego.
Hi, nice to meet you,
Diego,
Matt.
where are you from, Diego?
From Colombia.
From Colombia.
So glad to be here. Thank you.
Oh, mate, thanks for coming. Man, you're a fascinating guy and extremely bloody smart. We just had a little chat about what we want to talk about today, and there's just so much. So hopefully we can cover a fair bit. But tell us a little bit about yourself and your organisation and what you're working on.
Okay. So I'm from Colombia, in a city that is called Bogotá. It is my capital city of Colombia, and also I belong to the Universidad Distrital. That was where I just got my bachelor in chemistry. And after that, I started just moving for all of these biochemistry work. So I did sort of graduate programs and a specialization in systems biology and a master's in nutrition and dietetics. And currently, I'm studying my doctoral program in molecular biology and biomedicine in Spain.
Yep.
I belong
so you're studying in Spain?
Yes, but it's a partial. It's I work in the Colombia, but I move like 2 months or 3 months to Spain, so regularly.
Okay.
Yeah, because in Spain, I also work with an enterprise dietary supplements industry that is called MTX Nutrition.
Oh, yep.
Where I just act as the scientific consultant and that.
Excellent.
So that is so close for this University of the Basque Country, what it's called. Where I'm doing my degree. So the point is that I just met 5 years more or less ago, two very big professionals that were also from Colombia, another from Spain. And we just start doing some stuff together, and we consolidate what is right now called DVSS.
Yeah.
It's an organization of many professionals in different fields.
Yeah.
We have some nutritionists and dietitians, some trainers, or some others that are exercise physiologists or physicians.
Yep.
So what we do nowadays is to promote the public awareness of exercise sciences in Latin America and Spain also, and given the support and of many of these big associations and organizations as the NSSA or the ISSN. So we try to have some of these research projects with them, just to continue the developing of the country, because you know that in Latin America we have some lacks, especially economically, and some others maybe from the human point of view. So what we do is try to join efforts. So all of us belong to different labs. So we just do our best in every single lab and with the support of the others. So that's why we are moving a lot. So we have done right now conferences in Mexico, Costa Rica, Peru, in Colombia, Argentina, Chile. So it's great, the network that we have built. So
and you train people up, like they do courses through you, you've got educational material.
Yes, also,
So you use all the resources from all the best labs in the world, you network with all the best scientists, and then create all that data into training programs and information that you can take back through South America. And
exactly. So that's why we use our data that we are collecting from different studies that we started like 3 years ago. Just moving in all of several lines that we will discuss some. But also with the endorsement of the NSSA. So we certify the NSSA CPT, and we are working on the book, on the Dr. Kreider's book, will be used for the certification of the ISSN, the CE-ISSN. So
excellent.
So there are many staff that have been just propulsive with this network, and that we already have some background in graduate programs, because most of us maybe act as lecturers or research associates in masters and doctoral programs in Colombia and some other countries.
Wow. And you're publishing a lot of this data and everything as well. And what sort of projects you're working on now? Because you're telling me, remember, we just had some very brief conversations about some really, really exciting stuff. And I love the way you seem to be thinking outside the box with a lot of this information. Tell us what sort of projects you're working on now.
Yes, there are some projects that we have right now. One of them is the cluster training line. So the cluster training is like a cluster resistance-based training it's called. So this kind of approach is just like in a full set of 12 reps, for example. We divided in 4 clusters with 3 RM each one, and some intrapause. So with 20 seconds, it could be 40 seconds. So we already actually got some data that showed that the 20 seconds intrapause was better one. So that will be published soon.
So what you're saying is we do 3 reps. And what intensity? Are we going maximal, or like we do it only just do 3
RM? So with that, the purpose is to maintain the load, high loads. So that's why we use this kind of approach for trained population. And they currently just do it 3 RM. They rest 20 seconds, and they continue the other 3 RM. And so on until they got the full set of 12 reps. So because that's...
So do 3, wait 20 seconds, another 3, and you do 4 sets of that, and you hit 12. And is that, do you then do that again, or is that's that muscle, that's that exercise finished?
No, we normally use 3 sets.
So
of
3
each exercise. So for example, just to put it in a context now, like in 2 hours, I'm going to present in the data blitz here, the ISSN. Some of the data of this that we did was a cluster training program with in lower limb. So with leg press, squat, and deadlift. So they did 3 sets with 12 reps, 4 clusters with 20 seconds intrapause each one. So and we got 2 groups that were these cluster training. One of those groups intake 0.1 grams of creatine per kg and beef protein, half of body mass after training. So that was like the group, the supplemented group. And there was a control group also who continued the habitual diet and training and sort of. So what we found was better outcomes in strength, and also just doing the squat RM, the 1 RM, and also a trend in fat-free mass for the lower limb in the creatine group. So it was huge, the increase. Also, they were actually, where we were collecting the data, the increases was very good, impressive.
Is that right? So we're talking about building bigger legs. and burning off the fat by using the creatine in this style of training.
Yeah, and actually we divided because this is the data only for lower limb, but we are also working on the upper limb right now.
Yeah.
So and the guys are just responding similarly.
That's interesting.
So that it's one of the lines, the cluster training. So that as I mentioned it before, we already got some data about the intropose because that is something that we were like curious.
What's the theory behind that?
The theory is that given these small recovery phases that we have, so maybe the cell is going to recover faster the phosphagen system, and with that you are going to continue and obviously to remove some of these, avoid the accumulation of some stressors, as maybe hydrogen ions, some calcium, some others. With that small recovery, what we have seen is better outcomes in strength and muscle mass gains. So the trip.
So just for the listeners there, the hydrogen ions in Australia, we talk about acidity, and that's so what you were saying is by having these pulses, it gives a chance for the phosphagen systems to replenish, so you got more ATP available for the power exercise, and it gives a chance for the waste to be buffered and not accumulate the extra acid. Oh, that's excellent.
So and the first study that we did was to compare 3 cluster sets, because I guess that is the baseline, just to start just standardizing the method. So what we did was compare 20 seconds intropose, 40 seconds intropose, and one group who did 2 clusters only with 6 RM with 30 seconds between. So with those 3 groups and a control group, we ran some DEXA analysis, body composition, and also the strength with counter movement jump and the squat RM. So what we found was that there was better outcomes in the 20 seconds group. So that's why we move with this. Having said that, with these 20 seconds intropose, we start doing with some other protocols. Now we are studying creatine.
Was the creatine? Did the creatine have any effect on the 20 to 40 seconds or anything?
We didn't study the effect in those groups only. We just said like, okay, we're going to study from now on the 20 seconds. But the idea is to actually replicate in this network, because there are several professionals, as I mentioned, that they are working in their master's thesis or doctoral programs. So they are going to continue replicating the first part of this standardization of the cluster, but also to add some other things. So, for example, there are some coming in beta-alanine that we have in mind, and also because it's almost functioning with the same kind of approach, the system of recover faster, buffering. So
the beta-alanine, for the listeners again, that increases muscle carnosine, and carnosine, not carnitine, but muscle carnosine is, would it be the most powerful intramuscular acidity buffer?
This dipeptide is going to quench even some other ions and the hydrogen ions, and it's very popular nowadays. And the beta-alanine has a well-supported data in different studies. So that is the way that it works.
Excellent. Oh, that's really cool. What else? So you do a lot more with telling me more about this bioinformatics.
So that is some other line that we are already just trying to put some stuff together, because bioinformatics is an old discipline. It's like from 1960, more or less. So, but given this explosion of the genomic era in 90s and 20s, 20,000. So the 2000, sorry. So there are a lot of data that accumulates. So there was like a need to create more algorithms to analyze all of that data.
All the different genes.
Now we know. And not only genes, but also proteins, because you know that nowadays with proteomics, metabolomics, so it's huge, the amount of data. So what bioinformatics does is just to create some pipelines with computers where you can just add raw data to analyze it and to put it in make it sense. But that is only like a piece of this puzzle, because with bioinformatics, you just, for example, organize data from a sequencing. I don't know, could be that you are analyzing the differential express genes after a training session.
Yeah.
So with all of those data, so you need something, the computer, who analyze this. So
Yeah.
that the computer just given an outcome. But with that, we need to use the other approach, that is the systems biology,
Yeah.
where you are going to integrate all of that to a network. So, for example, in systems biology is an interdisciplinary field where you have this part, bioinformatics, but also the knowledge, the deep knowledge in biochemistry, and these low throughput that are called low throughput analysis,
Yeah.
Western blot, EMSAs, and some others. But they are called low throughput because you can run just one by one.
Okay.
But they can be mixed and complemented with the high throughput sequencing methods and some others, where you analyze the full nodes or the full entities of a network.
And can you use that? And what's the end goal of that? Can we use that to have a personalized prescription, or see what sort of training works for the person?
Yeah, that's my point. So, for example, you move from just the single methods, the low throughput, to the high throughput, and you combine them to create a network where you can analyze or to see with some other enrichment tools. You can see, for example, what is the pathway maybe which was affected the most by the exercise or some nutritional intervention or some supplements.
Yeah.
And with that, you can analyze the behavior of the network.
Yeah.
And obviously, that is something that was done, for example, in a paper of Safdar and Tarnopolsky in 2008, when they did an osmosensing analysis of all of the genes that change after creatine supplementation.
Yeah, right.
So, and they found different, not only mRNA expression profiles, but also the proteins, which kinases change after de-supplementation. And with that, they just got, after analysis, this kind of enrichment that I told you, what was like the main pathways that are affecting or being affected after the supplementation. So that's why they just got these MAP kinase, P38, or the sphingosine kinase, and some others that are more sensible for the osmo variation.
Yeah, okay.
So that is the point with the systems biology. So there is a need nowadays to continue developing more techniques. Actually, we are moving from the first generation sequencing methods that was just like by hand almost, just trying to sequence the DNA, for example, just adding step by step, letter by letter. So that's why it's huge, that it's difficult. So, but now the most common method that we use is the second one, the second generation techniques. They are more automatically developed, but there is a new one that is called nanopore sequencing. Those are the third generation sequencing methods. Actually, they have more advances in this one, nanopore sequencing, some others, OS4 methods that they are called.
So, because in Australia, again, not all of our listeners are in Australia. I shouldn't just keep focusing on Australia, but but they're the mainly people I talk to, I suppose. But a lot of people are getting gene tests, and they're telling me, I've got this genetic polymorphism or this SNP, or so. But we're trying to always talk about gene expression, and you've just suggested there that you've actually, there's technology available that shows that in response to certain training or certain supplementation, you can turn on or off genes. You can manipulate your genetic profile.
That's very important that you mentioned that, because there is a huge confusion between what is nutrigenomics and what is nutrigenetics. Because nutrigenomics is this field that I maybe have been talking about. Is the fact where you intervene in people with some of dietary approach or a supplement, and you see what is changing in the cell or in a given sample. But the other point is nutrigenetics. But that's very
individual, isn't it? That's unique to the individual.
Could be individual, or you can see it in a full sample. You can see how they have patterns. So we see maybe a regular, I don't know, a pathway or signaling pathway or whatever, or a gene that is turned on or sort of. But in the other case, the nutrigenetics is more individualized. But there are many funny stuff that appears nowadays because they are offering developing a lot of these methods for the like trying to have your personalized diet and that.
Yeah.
But that is in the infancy actually right now. All of those, because even the methods to analyze and the polymorphism and so on of those they came from saliva, and there are some like doubts about the efficiency of the method, but also not only that, but that this is more commercial, I see right now.
Yeah, okay.
Because there are some factors that obviously they can affect, for example, lactase deficiency because of some mutation or SNPs or whatever. So that can affect obviously the habitual diet of the person. But it doesn't mean that you need to have your full screen and of the genetic profile and that, because there are many stuff that are being just developed and continue to develop.
So and we even see people with what appears to be a good genetic profile expressing a defect in a genetic polymorphism because of the diet, lifestyle, or supplementation. So and conversely, we can probably change a lot of that by fixing a lot of these things.
Yeah, I guess that in future years there will be some new advances about this, but the technology must be improved, and also some of the methods that we use, the sampling and that, just to have a better outcome, because it's better to see not only these individual factors but also how they respond. So that's why it's better to integrate those two.
Yeah, exactly. And you want to see what's actually happening in the person. So let's look at what about microbiome? There's a whole, after that whole genome era that just went into this massive microbiome now. Is your work involving in with the microbiome?
Yeah, my actually one of the lines of the doctoral thesis is with analyzing the effects of creatine monohydrate in the microbiome. So how can affect different species and the characterization of that. So actually this systems biology approach is used to analyze metagenomically the microbiome. So what we do is normally we extract some samples, could be feces or could be from saliva, from different where the bacteria they are. So after that the DNA is extracted, and you have a portion that is called the 16S RNA. And there are some new methods to have some proofs, forward and reverse proofs in specific regions where you can have like the fingerprint of these bacteria. With that, you run you go reverse, like go for the phylogenetic analysis to maybe to find what is the bacteria which is the most abundant on that. So this has been developed. Also, maybe the audience could be interested in that. Dr. Jackson in George Winstock in the Jackson laboratory, they were having some results about the elite cyclists and characterizing the changes after how they have these different species of bacteria and some archaea in the gut.
So is there any particular bugs that thrive with the creatine?
there are like two in the with these cyclists. They were improved, the Methanobrevibacter and Prevotella, that were just the highest. But that can have some variations. Actually, yesterday here in the ISSN, Dr. Campbell was showing some results also from animal studies, where they found after some bouts of exercise, different changes also in microbiome. So with creatine, the point is that there are some hypotheses. In animal studies, there was one study by Langley in 2014, where they did in mice, like humanized mice, and they start giving some patterns of these changes after the aging process, and they found that the creatine and creatinine, the degradation product of creatine, were more metabolized during the aging process. So these bacteria, because they didn't analyze specifically which kind of these bacteria, because there are many. So what they found was that this aging process affects some of the metabolization of this molecule, the creatine.
So are you saying there's certain bugs that might encourage the conversion of creatine to creatinine before we absorb it, or was that?
No, is that normally we have discretion of creatine by means of creatinine in a non-enzymatic process. But the point it was that after the measurements that they were doing in some time points, they found more degradation in creatine and also in creatinine.
Yeah, right.
So because there are no.
I was wondering if the bugs were eating the creatine and excreting creatinine that we were eating.
No.
Because the bugs are going to get it first.
They are just metabolizing faster creatine and creatinine also.
Yeah, okay.
Because remember that they can have different enzymatic pathways than us.
Yep.
So they can use creatinine. We don't use it. We just excrete it. But they can use it. They can metabolize it to different products.
Yeah.
So there are several nitrogen derived products that came from the creatinine.
Yeah.
So that's why they were just more metabolized.
What about, I don't know, I'm just going to keep asking these questions.
No,
but have you noticed it with the plant, any vegan information, like plant-based, people on a plant-based diet, have they got a microbiome that can help them replenish their creatine or something? Is there any discoveries there? That would be interesting.
I'm not sure actually right now that there still maybe exists something like that, but there have been some studies nowadays. We are trying just to characterize populations and to that. Maybe in future years there will be some other better outcome to know which species is going to be better for given populations. But what is also known nowadays is the effect of some probiotics now. Some lactobacillus, rhamnosus, and some others.
Yeah.
The bifidobacterium, some families that can help and support some good ergogenic effects, but with some variability. So that's the point, just to find the better. Maybe that could be with a good outcome in the sports or ergogenic.
What about gut wall? Has it come across anything with the creatine in that gut wall? I noticed in the seminar they're talking again about heat shock proteins creating a leaky gut wall and that sort of stuff. And anything in there? Have you come across?
Yeah, but there are some anti-inflammatory effects of creatine also in these, actually measuring this HSP, the heat shock proteins, and some others. And for example, there is when there is a deficiency in the agate, that is one of the enzymatic steps of the creatine synthesis. The arginine glycine aminotransferase. So the people trying to have
that's involved in the methylation of guanidoacetic acid to creatine.
Yes, exactly. So in that step, when people have in some of these enterocytes, they have this lacking of the or low activity of this enzyme, the people, there is some correlationship to have bowel disease and syndrome. So that could have some good interesting profile.
Oh, it's fascinating stuff, eh?
So
because this is a big thing. We're learning so much now about the microbiome. We always looked at our body and looking at our own potentially genetics or whatever, but pathways. But the microbiome is going to get it first. So it's a really important and fascinating
and actually
topic.
Because we are like the host.
Exactly. But are we? I mean, there's 10 times as many of them there are as us.
We just might be
vectors for transport for them.
There is some data that says that nowadays there is like 1.3 or 1.4 bacteria per human cell. So they are more than us. So
I know. So they're probably just with you because they want to go to Cuba and Spain and then back to South America.
They have a very, good, interesting combination there.
Yeah, absolutely. So.
So that is one of the digamos, very nice topics that we have been working on. With the fact of different training interventions. And just using many populations, because we have been working for health. We are having a cluster training also for Asian people. So for older people.
Okay, it makes a lot of sense actually for that group too.
It could work because you optimize the job. You optimize some of
and they're more likely to get the injuries in that stage of fatigue as well. They start to lose technique and form. So with your cluster stuff, you're saying that you get that little 20-second break, because it might even be enough for them to be able to
it's not so
get the brain back into it and get that focus and even that muscle-mind connection, by just getting that time to just refocus and work on technique and form.
So the internal and external focus, actually, we try just to control it also.
So that cluster training, is that published data that people can find somewhere?
There are some other jobs already that have been done. And we already published one that was accepted for the ISSN conference. some others. So
I think our listeners are kind of
there are some that have been published, and there are some others that are coming.
Yeah.
And so in this same fashion, one other interesting thing is that we are working on the differences that can have these contraceptive pills in women.
Yeah. Talk to me about this.
Yeah, we found that using contraceptive pills doesn't affect the popular maybe thinking.
Yeah.
They are just concerned about the fat gaining or some other, or that decrease the performance and that, but we didn't find that actually
cool.
was similar increases. It's obvious that we need more control some other variables, because we mix some of these contraceptive pills, the monophasic and triphasic, in both groups.
Yeah.
So our next step is to analyze what happened with those girls who just intake monophasic and triphasic, just to see if maybe we have some changes. But at this moment, we didn't find any.
And were these any particular pills, like estrogens or progesterone-based pills? Or
these were just estrogens. Yes.
And no real link there at all with the changing in the.
No, with fat mass.
Yeah.
You said.
Yeah.
You mean? Yeah. No, there were no changes. We use Indexa just to measure the changes after the protocol. So was also a protocol. We already have some works before to optimize gains. So we use some protocol with from 8 to 12 reps. So the full series, like 24 sets per session, and with an upper and lower limb fashion.
Yeah.
So and with this method, and given that they didn't change them. And the other one that we were just talking was about the ketogenic diet. So we just published one article the last year.
Yeah.
And we found that in a hyper energetic diet.
So in a calorie surplus.
Mm-hmm, in a caloric surplus.
Yeah.
Trained men.
Yep.
They run this kind of approach of training, the same upper limb, upper lower limb.
Mm-hmm.
So just trying to promote a bulking phase.
Yep.
So we compare a high protein diet with the ketogenic and a high protein diet, but traditional. With the carbohydrates more surrounding the 55%.
Mm-hmm.
So what we found was that given this surplus in the ketogenic group, they don't gain muscle.
Okay,
They
so in a calorie
continue
surplus,
the
they
same.
weren't
The
gaining
lean
muscle.
body mass sustained.
Yeah.
That was maintained, but they lose some of fat. They lose fat, fat mass, and visceral adipose tissue. So it was strange. We found that. So
just so we got it. So the listeners got this right too, because I mean everything's calories in and calories out. I thought, and you go into a calorie surplus, you're going to grow.
Mm-hmm.
But doing a calorie surplus following the ketogenic style diet, they didn't grow.
Yeah, no.
But they actually lost fat.
Yeah, actually they lose fat. Yeah, and they maintain the muscle mass.
Yeah,
so,
wow.
Yeah, so was something that was strange.
Yeah.
That we found.
Was there any extra studies that, like looking at mitochondrial biogenesis or mitochondrial density?
Yeah.
Did something grow?
Yeah,
with that calorie surplus, was something extra building? Was density increasing, or was something? Or
I don't know. Maybe if for these other studies, because there are many, that there are some that promote the ketogenic, and some others maybe not. But
just for clarity, we're not talking about exogenous ketones in a normal diet. We're talking about
reducing the carbohydrate.
Intake. And the extra calories with them are coming from the fats.
Yes, sir.
Yeah, and the protein adequate.
Yeah, protein was like 2 grams per kg.
Where
it's supposed. Yeah.
Yeah. Wow.
So with that, we moved to in the line that we have now. We are replicating the same study, but in women.
Yeah, okay.
And the results until this moment, because they are not published yet.
Yeah.
They are even worse. Because the girls, they just lose strength and they are losing muscle.
They're losing muscle.
Yeah. So
and still losing fat.
Yeah, but some are still losing fat also. So there was like a reduction, because there are some many things and the people or some people are like debating, because the control of the diet that that this creates some satiation and sensation and that, but we control that we're using with several professionals that were behind.
So what do you think? What's your thoughts? What's your opinion of how this is
Because
happening?
they were trained people we feel like they are more adapted to use carbohydrate, and also that they didn't have, because that is something important, they didn't have ketogenic like experience or big experience before, maybe in the cutting phase and that. But that is not like enough to have like a good set of adapted genes or these kind of to metabolize fat more. Maybe that is some of the things that the people need to understand.
And the metabolic rate increases.
That they are more used to have these carbohydrates, and usually we recommend that because the type of exercise that we do during resistance training is more carbohydrate dependent. So that's why it's necessary to withdraw them. So and we are just trying to show that that is not happening. That is maybe it's not a good option in training population.
Because that's what we always talk about: calories in, calorie surplus or calorie deficit is going to determine if you grow them. But the maths is not perfect. How do you accurately calculate the potential calories in? We know with microbiomes alone could change your calorie yield from a meal by up to 20%. And then we're looking at the calories out, it's determined by metabolic rate and so many other factors. So.
Many factors. Actually, I would like just to quote Dr. John Arendt, his talk this morning, that he said that is just the energetic balance is good, but there are some other steps behind the when you intake the calorics, because there are many processes that happens before all of that energy goes into the cell. So that's why maybe you can have sort of a well-balanced energy, but that energy at the end then comes to the cell. So that's why maybe it's affecting some processes there. So the macronutrient manipulation and the timing is important. So there is some people that continue just saying like it's not important to intake, for example, or protein or carbohydrate after training, but all the science is supporting this. That is to have better supercompensation and to have better glycogen storage.
And it's all an adaptation game. So like the importance of cycling, you were saying before too, there's a very difference between an acute study and a long-term study when we start to adapt. So that's fascinating. I'm going to get those references and those papers and make it available when we post up the podcast so everyone can find this data and read it. I think it'll be fascinating.
Yeah, for sure. I will share it with you.
Was there anything else you wanted to mention, Diego? Because I'm conscious that you've got a lot on this afternoon. Because you're going to go out and win some data blitz thing.
With some data blitz.
Diego's got to go out and pretty much summarize a whole lot of work in 60 seconds. Probably what we did in half an hour, you got to do in 60 seconds.
But it's okay. But everything was very cool in that experiment, actually, because we find that the cluster training could be a nice strategy for some new methods. So we're
someone
going to
like me, I
that.
like it too. Like that idea of doing a bit and having a rest.
That's true. And my point is just to maybe to close, because I said so. Just to continue supporting interdisciplinary work. That everything must be based on science, and to have a better result is necessary to do what the science maybe have been studied, and to try when maybe some of these drawbacks that we have in science in some topics that maybe haven't been researched. The idea is to have some good coaches or experienced people who can help, and not to trust in a lot of things that you can find in in website advertisement, internet, social network, because that is putting worse things in here right now.
And everyone's different, so it's very important to take some sort of measurements. Get some sort of objective measurement so you know for you what's working, Because you'll find there's non-responders and responders for a lot of different things. You just got to know which one. I noticed in some of those slides today, I felt so sorry for some of those people because they were working. Man, these people were getting smashed training. Some guys were building muscle, others were just losing everything.
With that point, maybe it's important to know that it could be not only that they are just bad lucky, but could be a slow process. So could have a very slow process. But these can be optimized by some other supplements that, for example, in the study, they just did the comparison about the training fashion and that. But with creatine or with the carbohydrate good intake and the high protein diet. So those kind of, that are like our arms. Like the way that we can fight against these kind of non-responders.
Exactly. Oh, that's great. And so an individual approach is just because something works for someone doesn't necessarily it's going to work for everyone.
Yeah,
and that's
exactly.
an important thing. I'm glad you're there teaching the coaches and that sort of stuff how to actually do all this sort of thing properly, because there's no cookie cutter approach. There's no simple one way that works for everyone. So that's really fascinating.
Yeah, exactly. Yeah, and I would like just to maybe promote with Latin American people, this message goes from them that they need to change also. I mean, some staff in their minds to start doing more collaborations, because this is the way that they work.
Oh yeah.
That it works, because the more you have these kind of connections with excellent professionals as they are, so you are in constant growing and learning.
Yeah.
So, and that is important. And I mention it because these kind of different speed in the progression of the science or the economics in a country, it's depending on the mindset of a person. So
exactly.
that it's so important, and in my country it happens too much.
Yeah.
That the people
and you need to be open-minded to follow. And there's so much science happening all over the world, all at the same time. It's a waste of time if everyone's doing the same work. You need to collaborate.
Yeah.
And that's what you're finding. And then you can take it back to your people.
No.
In Australia, we're getting a lot of data from all over the world, and we need that to come back into us, and then we try to use our own resources to bring it all together. So that's, I think it's a perfect scenario.
Yeah.
Networking, that's what it's all about.
Yeah, networking is all. Yeah.
And especially with science, and this is where there shouldn't be and this is the other thing I've been really impressed with this seminar. You can have people with opposing opinions on the same data, and they're quite happily discussing why they both feel that same way, you know. It's very important to understand.
Yeah, that is something that we always
bias in. There shouldn't be bias in science. You should never take science personally. If something has been shown to do something in the past and then new data's changing, just embrace it. That's what it's all about.
Yeah, we try always to say that to the students and to other colleagues that we have, that we need to doubt of everything.
Yeah.
So we are doubting actually what we are finding, and that's why we continue doing more research on what happened if we change maybe this one, or maybe we didn't control this. So
and look for bias, you know. And like we keep saying, you got to share some of the bad news so we can further the scientific endeavours. I think it's fascinating stuff. That ketogenic diet's screwed. No, that's really messed with my mind, what you've just told me there. I think it's fascinating.
Yeah, it was fascinating, that one. Yeah, and the girls' results, and they are going to be more fascinating. We will see soon those published.
Yeah. So I mean, everything changes. You know, we just keep thinking we kind of think we know what we're talking about, and then we prove it to be slightly different. So no, it's good fun. Well, thanks for your time, Diego. I'm going to let you go and get ready for this thing now.
Yeah. Okay. Thank you so much just for having me here.
Oh, no worries. You got to get to Australia. We got to do. We're doing a summit later this year and bringing over. We're going to try to start doing them every year. So hopefully we can get an excuse to bring you over for one of them.
Yeah, that could be amazing. Yeah, for me, more than honored to be there.
Yeah, and bring your gut bugs with you.
Yeah.
Thanks, Diego.
Thank you so much, Matt.
And thanks for having us. Another
ATP
Project. Someone will finish it off.
Okay.